Shawna is a microbiologist and clinical researcher, and currently working in the PFL, but she did some work at SHUF previously. She's originally from the United States, but she's been living here for more than 10 years, and it's safe to say that she fell in love with this country. That's why she did her master, PhD, and she's still, and even working at Nestle. And she's really in love with the mountains. She likes to talk about science, politics, and she also loves Thanksgiving, and she'll talk to us tonight how viruses can cure bacterial infections. So please welcome Shawna McCallin.
Good evening, everyone. Can you hear me okay if I talk like this? Yeah? All right. Thank you, Dimitri, for the introduction. This is a really great event. It's the first time that I'm actually attending Pint of Science, but will definitely not be the last. And I'd like to thank all of you guys for coming out. It's a Monday night. It's a rainy Monday night, and it's a rainy Monday night when there's the final season of Game of Thrones. So thank you for caring enough about science, for being here. And with that, I'd like to start off and tell you a little bit about how a spoonful of viruses could help us to avoid an antibiotic apocalypse. So this is a subject that I've been studying now for 10 years. It started off when I was sort of a snarky bachelor student and didn't really know what I was doing or if I wanted to do research. And then I fell in love with bacteriophages, and 10 years later it's still, it's my career and my passion. But before I talk to you about that, we're in a pub. There's nothing better than pop quiz trivia for a pub. So that's where I'd like to start off tonight. So we have a couple of questions. We'll go through them one by one, and I do know there are some microbiologists in the audience, so if you know the answers too quickly, hold on. But I want to know 5 names of bacterial species, so both the genus and the species name in Latin, please. Then I'd like to know what causes pneumonia, then what do vaccines do, and what do antibiotics do. So let's start off with the first question. And if you have an answer, go ahead, just yell it out. Everybody loves to yell bacterial names really loud in bars.
Staphylococcus.
There we go. Staphylococcus, what? That's just the genus. All right, so we've got that. Anybody else?
E. coli.
E. coli, yeah.
Yersinia pestis.
There we go. That's a good one. That's an old one. Haven't heard that one for a while. So there are loads of bacterial names, and there's some that I'd like to bring to your attention more than others tonight. So the ones that I'm going to talk to you about, or what my talk really concerns, are called the escape pathogens. So these are the ones that we're having a really difficult time with treating in the clinic now. So escape stands for Escherichia coli, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterococcus faecalis. So these are the bacteria that I will ask you later about to see who remembers, but these are the bacteria that we're really having a hard time with for treating with antibiotics. And then, being a microbiologist, we get really used to not actually seeing what we're working with, right? Micro, super small. So if you have an idea of just how small a bacteria is, if you could imagine this big black circle is just the size or the diameter of one of your hairs. So that's a really big hair right now, right? So that's the size of one of your hairs. Now, if you look at that, that is the size of one of your red blood cells, proportionately. And if you can see from way back there, wait for it, there it is, that little red spot is what a bacteria would be like. So it's really, really tiny. It's about 1 micron in length, or micrometer in length. And that's really what we're working with. And luckily, we've got really good microscopes. And if you want to see what some of the bacteria look like, we have these amazing photos like this. So this is Escherichia coli. I mean, I think it's pretty much a work of art. It's beautiful. But it also can kill you. That's how bacteria work. So this is, if we go from all these different names, they're super small, and then this is how we can actually be able to see them. So that's the first question. 5 names of bacteria. I don't think any one person gave me 5, but there's chances to win a free beer. I will give you a free beer afterwards. So what causes pneumonia? So first of all, pneumonia is this disease, right, where your lungs get inflamed, the air sacs aren't, doing their job, and breathing is difficult, and it's not a good condition. Anybody here have pneumonia before? Yeah, me too, during my bachelor's degree. All right, so some people understand pneumonia is not nice. So what caused your pneumonia, if you don't mind me asking? Whoever had pneumonia?
It's basically a family of viruses.
So he had viruses. What did you have one too?
No idea.
Okay. So he says viruses. What else can cause pneumonia? Anyone? Come on, microbiologists. Bacteria? All right. Yeah, so we do. There's bacteria that can cause pneumonia. There are viruses that can cause pneumonia. And there's fungi as well that can cause pneumonia. So this is the idea that there's conditions that we have that can make us sick, but we have to be really sure actually which one is causing it to know how we can treat it. So then that brings me to the next question about vaccines. So vaccines are one of the tools that we have against microbial infections. And so what do they actually present? We've got A, bacteria, B, viruses, C is both bacteria and viruses, or D, none of the above. Who says A? B, C, and D. All right, good, thank you. The answer is actually both A and B's. We had a mixed response between either just viruses or viruses and bacteria. So vaccines, virus, they kind of sound alike, but they're actually used to treat both bacterial and viral infections. And just to remember how they work, so you know those are the shots we get a lot of times when we're young or when we go to the doctors. And what you're actually getting injecting with is little pieces of the pathogen, so pieces of the virus or pieces of the bacteria, and that stimulates your immune system to make antibodies. And then these antibodies are actually what fights off an infection. So when you get sick, it's actually your own body that's fighting the virus or the bacterial infection. And this is the type of strategy that we use to prevent infections. So if you're already sick with pneumonia or whatever else you might have, it's not the right time to get your vaccines. So make sure your vaccines are up to date. Now, examples of that are for the flu, for rabies, tuberculosis, smallpox, HPV, and now there's even one for malaria, which is a parasite. There's a lot of different vaccines out there. Make sure you're up to date. But then what happens if you don't have a vaccine and you get sick? Normally you'll go to the doctor and they'll give you antibiotics. So same question, what do antibiotics treat? Who thinks bacteria? Who thinks viruses? Who thinks both viruses and bacteria? Oh, good. Good. I don't have to go over this. So, antibiotics are to treat bacterial infections. So again, this goes back to why if you have something like pneumonia, it's really important that we know what's causing it, to know how to treat it. So antibiotics kill bacteria, and so that means when you get sick with it, it's not you who's fighting off the infection, it's the chemical antibiotic that then attacks the bacteria within your body. So this is something you can take during treatment or just before you get exposed to bacteria. That's when you take it. And then this is just a nice image to sort of remember where we actually got antibiotics. Antibiotics were discovered in 1928 by Alexander Fleming because he had these petri dishes. And then he noticed that there were some contaminations. But he was quite a curious scientist, so before just throwing them out like I do when I get contaminations on my petri dishes, because it does happen, he noticed that if you can see here, this is where the bacteria grow, then here's the contamination, and then there's this halo of inhibition where actually nothing grows, and that's because the contamination of the mold was releasing penicillin. And that's where our first antibiotic came from. So that's antibiotics. And that's our pop quiz, except for one last bonus question. And if somebody can give me the right number, this is where you get a free beer. So it takes a little bit of a morbid turn at this point, huh? I would like to know how many people died in Europe in 2017 due to antibiotic resistant infections. Anybody have an idea? Anybody want to guess for a free beer? You guys are quite a bunch tonight. So who says less than 1,000? Less than 1,000 in Europe. Who says 20,000? There we go, couple. Who says more than 50,000? All right, so okay, we've got some ideas of how many that might be. And if we look at antimicrobial resistant deaths in just 2017, so in the world there were 700,000. There were only 25,000 of those 700,000 in Europe, which is doing good, so that was the answer to our question. 25,000. So whoever said around 20,000, if you come up to me later, I will give you a free beer. And just to bring that a little bit more closer to home, there was actually 300 people who died in Switzerland, which actually doesn't seem that much when you look at the bigger number, right? 700,300. I'd say bravo, Switzerland, you're doing well. Good job. We're actually very lucky to live in a country that does so well with antibiotic stewardship. But if you look at those 300 infections, or deaths, they were represented by 7,000 infections. And we have this measure that's called a disability adjusted life year. And that means that, hold on. All right, so this was supposed to show as well that there are 7,000 infections that represent a lot of time off of people's lives. So it's only 300 people who died, but 7,000 people who are infected. And now when we look at those numbers, now it's actually fairly manageable. 700,000 in the grand scheme of things of the world is not a whole lot. But there's a lot of projections that are going on right now to say that if we don't do anything about antibiotic resistance, that from now until 2050, that's only 30 years away, that there'll be 10 million. So that's 10 times as much as what we're dealing with now. And that's actually more than the entire population in Switzerland every year, and more than the number of cancer or deaths that we have to deal with every year. So that's a lot. And that's where, you know, a little bit dreary and sad at this point, but that's when science comes into play. And science is this is why I got into it, is this idea of what can we do and how can we address some of these problems. So for me, when I'm a scientist, the two things that I need to be is that, first of all, is I need to be curious. It's one thing. You want to have to know what things do and how they work, and figure out how you find out the answer to those questions. And then you also need to have hope, which sounds like a funny word that we don't really hear put in relation with science all that often, is, you know, you have to have hope. But for me, it's the fact that what I do it because I hope someday this is going to do something good. This is going to be useful for us in the future. So that's where it comes into me. And if I look at the subject that I really study a lot, it's this alternative to antibiotics called bacteriophages or phages. Who here has ever heard of a phage? Wow, this is the first time that's ever happened. So pretty much everybody's heard of a bacteriophage. That's great. So I won't have to talk too much about the details of phages and their structure. They've got this nice capsid or head that contains the genetic material. They've got a tailed tube, and this is pretty much a tube for the DNA to pass through. And then they've got these tail fibers, which are super important. We're going to come back to in a second. So phages, they're inherently a virus, right? So now I've told you that viruses cause pneumonia, and that I'm going to give you a spoonful of virus to cure your bacterial infection. Sometimes that sounds a little weird to people, but actually the reason that those two ideas are not incompatible is because of these guys here. So when we go back to talk about these tail fibers, you can think of them a bit like the feet of a bacteriophage. And the feet of the bacteriophage need to find shoes that they can go into. So when you look at the relationship between a bacteriophage and its host bacteria, it's very much like a Cinderella story. So here you got a phage, and she goes around and has her feet, and putting her feet into all these different shoes until she finds a match. So this is why these type of viruses are super specific to bacteria, not even just to a bacteria, but to the species level. So if it's a phage for E. coli, like the bacteria we saw earlier, and it comes with its E. coli feet to a Staphylococcus aureus shoe, they won't match. So this is why phages are extremely specific for bacteria to the species level. And then they have this simple lytic cycle of infection where the phage will come, it'll absorb to the surface, so the shoe and the feet mechanism will match. Then they will sit down and eject their DNA from the capsid into the intracellular space of the bacteria. At this point, it sort of takes over the host system, and it reprograms the bacteria to become a viral sort of zombie factory, and creates all these new viruses. So this is the stage of replication. And then the lytic life cycle will terminate when there are these proteins that are expressed called the wholin and the lysin, and this will cause the bacteria to lyse and then release a load of different phages that it produced. So here you start off with one phage in the beginning, and sometimes you finish with 10, 200, or sometimes as many as 1,000. And each of these new baby phages can then go on and set up its own cycle of infection. So you basically have a self-amplifying medication. It's very different than the way that antibiotics work. So if we go back to bacteriophages, this is not a new concept, really. I mean, it seems great so far. We've got antibiotic resistance. There's these viruses that, I mean, they're in the nature, they're in the soil, they're in the lake. They're everywhere, but somebody else had this idea long before we did. So bacteriophages actually were discovered in the early 1900s, 10 years before antibiotics. So this is Félix Derel, who was a French researcher. There was another guy who kind of helped out, but he studied phages a little bit and then went his own way, whereas this is sort of like the father of phage therapy. And he traveled the world, which was not particularly easy to do in the early 1900s. But this man went to India, Egypt, South America, and he was spreading phage therapy for different uses like dysentery or cholera. And then he met one man named George Eliava. Now he was from the Soviet Union, he was visiting in Paris, but they went back to what is now present-day Tbilisi in Georgia, and they built the Eliava Institute. And this was from the 1920s up until this day, is the cradle of phage therapy, where they produce phages and they treat patients, and it's part of everyday medicine. So if you walk into a pharmacy, say in Moscow or in Tbilisi, you might see something like this. So these are different phage products that are produced either for different clinical indications or for different bacterial species infections, and you can actually buy them off of the shelf without the need of a prescription from a doctor. And this is where my research comes in. All right, so title's gone, but that's okay. So this is the idea of what is actually in a bacteriophage cocktail. And I can tell you I've got one in my pocket. This is intestiphage. The photo up here is pyophage. So if you are interested in seeing what they look like later, I've brought some that I've bought myself here. So pyophage is a product that is against pyogenic infections. So typically wound infections, fever, pus, swelling, all that kind of gross stuff. And it's for any type of infection caused by staph, strep, proteus, E. coli, or Pseudomonas aeruginosa. But the thing is we don't actually know, or we didn't actually know, what was in these vials, because this is something that they've been producing in Russia and Georgia for nearly 100 years now. So what I did is I used different types of tools, because let's face it, in microbiology, your 5 senses don't get you far. You need a little bit extra help with that. So I first used a microscope, and through this I was able to see what my phages looked like. So using electron microscopy, this is what some of the phages were that are in here. You can see they're all with the long tail. You can kind of see the tail fibers here. And here you have at least 4 different shapes of the phages that are in the product. Then I also used DNA sequencing. So you can take out all the DNA of all the phages and do a technique called metagenomics. This is basically where you suck out all the DNA, cut it up into a bunch of little pieces, sequence it, and then try to put it back together like a massive jigsaw puzzle. So luckily we have a lot of computer power to help us with that, or else I'd still be doing my thesis today. And through metagenomic sequencing, I was able to assemble, I'd say around 20 or 30 different genomes that are present in this vial. So they have at least, I'd say, 20 different types of phages in here. And what you're looking at here is each bubble represents a different genome. The size represents how prevalent it is in the overall composition. And then the color represents what species it would attack. So the yellow one, for example, is Staphylococcus aureus. So in this product, there's one major phage against Staphylococcus aureus, and it's super present. Whereas you look at, in green, that's Pseudomonas aeruginosa, so there's at least 3 that are highly present, and 2 that are very, barely detected. So this is what I did with metagenomics, to have an idea of what viruses are there and how, in what type of relative proportions. And lastly, we have a lot of in vitro assays where we can check the actual biological activity of these phages. So here is what's called a drop test. Anywhere that's white is where there was a bacteria, the bacteria was growing. And then all of these spots represent where I put little drops of the phage product to see if it would lysis. So that's what we're looking for. We're looking, does the phage actually lyse the bacteria? So you can see when it's clear and green, that means that the phage was active against that species, or that strain rather. And then when you have in red, that means that that phage didn't particularly have any lytic activity. So this is how we know which ones could be useful for therapy. So if you're a patient, you get sick with one of these bacteria, you'd say, oh, I'll take that one there, maybe not that one. So those are the type of things that I would do to try to understand what these phage products were like. Now, if something's been around for 100 years and we're dealing with antibiotic resistance, there's a lot of people working on this subject. A lot of really great researchers, doctors, institutes. And this is sort of a map that shows the activity across the world. But for those in the back, hopefully you can see this a little bit easier. So in red, we've got countries where it's approved use. And this is a lot of countries from the former Soviet Union, in particular Russia and Georgia. And that's where they can use them as an approved medicine. Then we've got 3 orange dots, which represent experimental treatment centers. So it's not approved in these countries, but they're doing it on a fairly regular basis, either within the hospital or within the country. So we've got one that's actually in Belgium. Poland has been treating people with phages since the 1970s, so they have a lot of experience in there. Belgium has actually only been about a year and a half since they've been able to set up a sort of legislative alternative framework. And then there's an experimental treatment center in San Diego. Now there's a lot of other people that have actually been treated with phages for compassionate means, so meaning that the antibiotics were failing and then their doctors or the researchers got special permission to use phages as an approved therapy. So all of these yellow dots show where there have been cases across the world, and there's been at least over 30 cases of that happening across the world, anywhere from Washington State down to Australia. And lastly, there have been some clinical trials. So there was a clinical trial done in Bangladesh, so it turned out to be orange too. So Bangladesh was actually a trial sponsored by Nestle, which is something I worked on. Then there was a clinical trial for burn wound patients between France, Belgium, and Switzerland. And then there was also a trial that was done 10 years ago in the United Kingdom. So this has been something that isn't new, that we're still working with. And then you might ask yourself, then why don't we just have it, right? So we need something, we need an alternative. So why hasn't it become a reality for us in Switzerland or in Europe or in the United States? And that's because if we go back to what science is really about, yes, I'm super curious and I've got a lot of hope for phage therapy, but nothing in science is supported without data. So data is really the underlying factor with what we haven't been able to procure for phage therapy yet, and what we're working, because part of the drug development process, it's a long process, haven't been very convincing. They've either been very small populations or they haven't shown any sign of efficacy. So this is where we need to get over this hurdle of phase 2 to understand if the data is showing that phages are actually treating patients or not, before we can move into getting a regulatory approval. And we've got regulatory approval here in Switzerland. We've got Swissmedic. The European Medicines Agency is the governing body across Europe, then the FDA, or the Food and Drug Administration in the United States. And there's other per each country and different regions of the world. But that's ultimately what you need, is you need a regulatory approval before you can get to patient care. And so that's really what I've kind of now dedicated a lot of my research efforts to, is in how do we take something, this bench to bedside paradigm, when you have a good idea and there is a need, how do we get there? And it's a long and frustrating process. But the things that I'm really working on now is a biologically grounded regulatory framework. So we don't have any other things that exist like phages into our system. So our regulation right now isn't very well adapted for trying to trial these products or regulate how we can change them or not. So when I say biologically relevant, I do things like this, where I'll look at the proteins that are released by a bacteria, for example. This is a bacteria that grows normally, and the proteins that are outside. And then if I grow it with phages and sort of explode it, these are the different proteins that we see. So for me, that's more biologically relevant, right? What type of proteins are released from a bacteria after we treat with a phage? So these are the type of data that I'm trying to generate to say this would be important if we are going to develop a new framework. Then I'm really interested in looking at different ways of clinical trials. So we sort of have the standard paradigm of superiority trials where we compare, say, phages to an antibiotic or a standard of care or even placebo. And so far we're seeing really miserable results. So I'm working to try to understand what else could we do, or how else could we structure patient populations to actually see if there's a signal of efficacy or not. And then lastly, I'm really intrigued by this idea of artificial intelligence. So I'm a microbiologist. I don't really do a whole lot other than metagenomics, but there is a startup company in Yverdon who's called Phages 4A, and this was based actually in an FNS funded project. And the idea is that they can use artificial intelligence, where we have information about phage genomes, bacterial genomes, and then lysis data. And if we put that all together and let the computer sort of think about it, the computer will be able to say, if we have a bacteria from a patient in a phage bank, which one of those phages could be used for treatment? And that might sound like an arbitrary thing, but sometimes a phage bank has 10,000 phages in it. So you can either go one by one and do those 10,000 drops, or we can see if artificial intelligence can help with that or not. But I think there's a lot of potential behind that idea. So this has been a bit of my dream, to be in a bar where people have to listen to me talk about phages. So thank you very much. It's a subject I really love. I would like to thank a lot of the people who I've worked with, from my PhD to SHUV to Phages for All to Nestle. I've worked with a lot of really great people who are also so passionate about this subject. And I've also been really lucky to have some additional funding to help me continue my research. At this time, I would say if you are interested, there are a lot of options out there. If you want to know more, either tonight, I'm very happy to talk to you about it more. But otherwise, there's a lot of different things. There's a TED Talk. I think it's called How Superbugs Saved My Husband or something like that, something from sewage. And this is a really great talk about a woman from San Diego who her husband fell sick and they treated him with phages. And they also wrote this book called The Perfect Predator, if you need something for your summer holiday to read. Also more locally, there's the Epruvette, which is a public laboratory at the university, and I also work with them, and they've established a program called PhageBack. So if you would want to come and do any of the experiments yourself, we'd be very happy to have you there. And just to finish with, there was a recent case, this only came out 2 weeks ago. So here is a 15-year-old cystic fibrosis lung transplant patient who was basically on her deathbed. She had palliative care, they stopped antibiotics, and that was it. I mean, this is not what you think of for your 15th birthday. All of us here remember our 15th birthday, and it wasn't that. So they found a way to get phages. So she had an infection caused by a Mycobacterium species. And actually the same laboratory where I started off as a sort of lazy bachelor student over 10 years ago is the one that produced the phages. And now this girl is healthy and hopefully she'll live a very long, happy life. She got phage therapy, I think, for 12 weeks intravenously, and it worked really well for her. So, and it was also the first time where they used genetically modified phage. So it's just to show that this is a really relevant topic for right now, and hopefully we'll see this transition from bed to bedside so that when we have another 7,000 patients maybe in 2020 in Switzerland, we'll have something else to offer them. So thank you very much for your attention, and if you have questions, I'd be really happy to answer them.
Yeah, thank you, Shawna. So we're open to some questions. Gentleman over here.
Hello, Shawna.
Hi.
Do you deactivate your phages in this cocktail, like other pharmaceutical company deactivate bacteria to produce some drugs to prevent?
So you're talking about like almost like a vaccine, like?
Not like a vaccine. They deactivate one group of bacteria with thermal treatment.
Uh-huh.
And then you need to take it like a normal drug, and it prevents some not pneumonia but broncholia.
Okay, not to my knowledge with phages per se. So there's a lot of different ways we can use phages. So we can either, it's always to target a specific bacteria, right? And so for that, what we've only been seeing recently is really acute treatment. So if you've got a patient who's very close to losing his life or in a very dire situation, that we choose phages that are specific for that infection. As far as deactivating them, no. I mean, for the tests, like when you would do a test, sometimes you would deactivate the phage and then try to see if it still had an effect. Some people think that phages have sort of a vaccination-like also property to them, but this hasn't been something that they use in terms for treatment, no. Thank you.
Okay, I forgot to ask, what is your name?
My name is Mark.
Thank
Mark.
you, Mark.
Okay. Next question? Yep, there's another Mark here.
Hi, thank you for this talk. Can you treat with phages a patient which is chronically infected with the same bacterial species or not?
Yes, yes.
Because I get that the phages are recognized by the body like a foreign antigen, so you produce antibodies against the phages. So if you receive the same phages over time, the phages will be attacked by antibodies, right?
Yeah, so this is actually a really good question, because anything that is foreign in our body's going to recognize and eventually create antibodies against. It's still an area of investigation, but like the case that I presented towards the end there, she was treated for over, I think it was much longer than 12 weeks actually, and they tested the reaction of her sera to the phages afterwards. There's a general trend that you will get some level of what's called neutralizing antibodies, so it might help with clearance of the phage after a certain amount of time, but the bodies will not attack, or our bodies will not attack a phage like it's a pathogen, the same way that it would with a bacteria. So we don't have as much of a risk. It's still something that we constantly look at over time, but it shouldn't create that much of an issue. And then another thing you can also do is the libraries of phages that are out there is, you know, if you would have an issue with one, a bigger issue is actually resistance of the bacteria to the phage. And if that happens, then you just change the phage for another one. So if you get something that's protein is very different, you shouldn't have that same reaction as well.
And I think this little girl were treated with 3 different phages at the same time, right, to prevent...
Yeah.
Okay. And I have another question, if I can.
Sure.
Can a company like Nestle can patent a phage or not? Because I heard that it was not possible to patent a living.
It isn't. It isn't possible to patent an individual phage, but it is possible to patent compounds or mixes. So if you have the right idea to put exactly these 10 phages together, then yes. There's a lot of issues in terms of patent protection with phages, so this is one of the issues that's sort of slowing down the development of phage therapy, is because yes, you can patent it, but actually the genius behind phage therapy is that you could change it. But as soon as you change it, then you lose patent protection, and then you get to start that whole process over again, which is very expensive. So this is part of the issues. Is technically, yeah, you can patent a composition or a cocktail. You can't patent the ones individually, but if you modify them genetically, then yes, you can.
Okay, more questions?
There's a question in the back.
Miriam, on the back, question? Please don't forget to introduce yourself.
My name is Chris.
Hi, Chris.
Hi, sorry, maybe you said already, but I think you mentioned that with regards to the phase 3 clinical trials, there was only like 3 or so?
Phase
Is that correct?
2. Phase 2.
Phase 2?
Yes.
So nothing went to phase 3?
So there was only, out of the 3 trials there were, only 1 showed signs of efficacy. They actually stopped the trials so early because of how much of a sign of efficacy they saw. So this was for treatment of chronic otitis in the ear, Pseudomonas aeruginosa infection. This was done 10 years ago. They stopped the trial early with the recommendation to move to phase 3, and that's never happened. So this is part of the problem of small biotech companies. So phages don't really, on a normal basis, they don't interest big pharma. So you've got small biotech companies that don't have the means to do clinical trials. Clinical trials are so expensive now. So what happened with this small company is that they did the phase 2, they saw the signs of efficacy, and then subsequently, I don't know the exact details, but they got bought by another company. And then, if I am speaking correctly, then what it was is that they didn't see the market for it, so the long-term market. So maybe there's not enough people who are suffering from chronic otitis with Pseudomonas aeruginosa infections. But it was the only time that we saw a sign of efficacy. The other 2 were diarrhea, this didn't, E. coli diarrhea, and that didn't work. And then phagoburn trial is for burn wound infections. There are some issues, and both the trials, there's reasons beyond the phages themselves that explain why they failed, but ultimately they failed.
But you said in that one trial you had efficacy.
Yes.
Why would then companies not want to fund that if it's good enough?
Well, you can still fund it, but you have to think about your return on investment. So if you fund a phase 3 trial, which is financially significant, you have to see sort of on how many years you can actually make back that money. So maybe if you don't have a lot of people suffering from chronic otitis caused by only Pseudomonas aeruginosa, then you look like, yeah, it does work, but we're only going to get 10 patients a year. So we're going to put millions and millions into doing a phase 3 trial to make back a couple, I don't know, 100,000? And it's an unfortunate way to think of treating people and people's well-being, but it's also a part of a reality. So this is one of the things we're looking at, not just a different type of clinical trial structure, but who's going to pay for them. The most recent clinical trial was done between Switzerland, Belgium, and France, This was funded by the European FP7 framework, so it was grant money. And this is something that phage researchers and for clinical uses are looking more for grant money to be able to get to that level before they ask for financial investment.
Cool. Thank you.
Sure.
Thank you very much, Shawna.
Thank you.